Hello, my name is Shawn Shafer, and I'm the Aldevron Genome Editing Platform Lead. I'm going to be speaking to you today about the work that we've done at Aldevron to manufacture CRISPR RNPs for clinical applications. And so when I joined Aldevron, they were already offering GMP-Cas9 for CRISPR therapies. And in the last 2 years, we recognized that there was a great need for having a comparable product around RNPs. And so I'm going to talk today about some of the work that we've done to establish that. So to understand this, first you have to know a little bit about CRISPR. Since CRISPR is pretty prevalent these days, I won't go into much detail, so I assume most people are familiar with it. But it is worth establishing that the RNP is the ribonucleoprotein complex between the Cas9 protein and the guide RNA. And so here's a picture of the RNP in the cell. You see the Cas9 protein silhouetted here in the background, and it is complex with the guide RNA as it binds to the genomic DNA target. After this binding occurs, these two nuclease domains, represented by these little silhouette claws here, then bind and cut the genomic DNA. And this is the process by which researchers inactivate or knock out a gene using CRISPR. So one current and important example of how RNPs are being used in the clinic today is in the field of immuno-oncology. And so in the last couple of years, the first generation of CAR T therapies have been approved in the form of Kymriah and Yescarta. And these are autologous T cells that have been modified with a lentiviral cassette expressing a chimeric antigen receptor against the CD19 antigen. And so in the last few years, however, it's been demonstrated that knocking out endogenous T cell genes can greatly enhance the therapeutic potential of these T cell immuno-oncology approaches. And the same approaches are being applied to other immune cell lines. But really, CRISPR RNPs are the safest and most effective way to knock out these genes. RNPs circumvent the need for transcription and translation, so they're immediately active upon delivery to the cell. And importantly, they also begin to degrade immediately, so greatly reducing the off-target potential of the RNP. In the table at the bottom here are some examples of native T cell genes that have been targeted in the literature to accomplish an improvement of the therapeutic profile. So some examples include T cell receptor genes, the PD-1 gene, and then HLA genes, which have been targeted more recently in an attempt to create allogeneic cell lines. And so this is just the tip of the iceberg, and this really only represents the use of RNPs in immuno-oncology. But again, I think this is an important and immediately relevant way to look at the significance of RNPs and the need for a path to the clinic for an RNP service process. So with this in mind, Aldevron set out about a year and a half ago to establish the optimal conditions for complexing and characterizing the RNP. And so some of the important parameters we thought to define were first, what are the ideal conditions and ratios for complexing the guide RNA and the Cas9 protein. We then needed to create an in vitro cutting assay to be able to determine relative cutting activity of individual RNPs. And then in an effort to look at RNPs in the same way we would a drug, we wanted to really be able to understand the context of the RNP cocktail. And so to do that, we needed to really be able to accurately identify the RNP content relative to the free Cas9 or free guide RNA content for any individual RNP. And along with this went the creation of a QC panel and some standardization around RNP creation. And then importantly, really making an effort to understand the long-term stability conditions to store RNPs at so that we could extend the shelf life and treat them much more in a drug-like fashion. And so all this work culminated in this poster over here. This is really the highlights and the summary of the final assays. But today we're going to go into a little bit more in depth at how we arrived at some of these conclusions. So our first step was determining the optimal complexing conditions of the guide RNA and the Cas9 protein. And to accomplish this, here is just an example of one of the RNPs we assayed. And so in the starting on the left side of the gel here, these are increasing ratios of guide RNA to Cas9 protein, beginning at 0.5 guide RNAs per protein molecule and increasing all the way up to 1.5 guide RNAs per protein. And so for those of you that aren't familiar with this type of gel assay. It's pretty straightforward. This is a linearized plasmid that contains the guide RNA target sequence. And when it is incubated with the RNP, you get asymmetrical cutting, resulting in these two bands on the bottom here. And so with a robust RNP, what you would like to see is a diminishment of intensity of the top band and an increase in intensity of the bottom two bands. As we move left to right in this gel for each specific ratio, there is a decrease in RNP concentration. And so you see that the highest concentration on the left here establishes robust cutting, which then diminishes as the RNP concentration declines. So you might notice here, while you get some level of established cutting at the 0.5x ratio of guide RNA to protein, when you increase that ratio up to 1.5x, you really maximize your cutting potential. And these findings were also borne out in cellular cutting assays where we saw similar results, which we will not review here. Now that we have looked at some of the optimizing of the complexing ratios, we wanted to move on to establishing an in vitro cutting assay. The goal of having this assay initially is to simply ascribe the ability of cutting or no cutting to the RNP. A longer term goal is to be able to correlate what we observe in the in vitro cutting assay with what's observed in cellular cutting assays for a specific cell line. Doing this kind of work will take many more RNPs and a much greater range of cell lines to be assayed, but this is something that we think will be very useful for the field going forward. In the meantime, when we look at the bottom gel here, our starting point was to find a linear cutting range and the limit of detection for an in vitro RNP cutting assay. So again, here on the far left, you have the highest concentration of RNPs diminishing down to 0 on the far right. And so as we just discussed, you see a reduction in the intensity of the top band corresponding to RNP cutting activity that then declines as the concentration diminishes, with a limit of detection of around 0.01 picomoles in this assay. And so looking at our graph on the left, we establish a nice linear range of cutting activity for this specific RNP, and this is what we use as the basis for our in vitro cutting assay going forward. Now that we've identified optimal complexing conditions and a way to assess cutting activity in vitro, our next step was then to be able to establish the content of the RNP mixture. So there are 3 possible populations. There is the RNP itself, there is free Cas9, and there's free guide RNA. We began by focusing on how to measure and assess the free Cas9 in the mixture. We started by looking at differential scanning fluorimetry, the graph on the bottom left, and what we found was that we could not establish a linear range for detection of free Cas9. We then moved on to size exclusion chromatography on the right there. And once again, we found that based on size, that we were not able to tease apart free Cas9 from the RNP complex as they elute at the same time. It was then that we moved on to analytical cation exchange chromatography. And so here on the far left, we began here hypothesizing that the complexing of the protein to the guide RNA might increase the negative charge of the RNP complex relative to the unbound guide RNA. And that was borne out in our assay results. So in the far left, the top chromatogram, you see free ApoCas9, which elutes at around 14 minutes from the cation exchange column. And then on the bottom left, you see the RNP, which elutes around 7 minutes. And so we established on the right here that when run together, that after optimizing buffer conditions and column conditions, that we were able to get distinct separation of the RNP complex from free Cas9 on an analytical cation exchange column. Importantly, in the process of conducting and optimizing these assays, we found that the quality of the guide can significantly impact the ability of the cation exchange column to distinguish between the free and the bound Cas9. So when you look at this gel image on the left, in the first lane next to the ladder, you see a highly purified guide RNA. As you can see, there's a tight band and a few other products in this specific gel lane. When we look at the 5 lanes to the right, these are lower purity guide RNAs, and you can see there are quite a few more additional products in the content. When we complex these guide RNAs with the Cas9 protein and run them on the cation exchange. In the top right here, you see the high purity guide RNA. And once again, we're able to clearly distinguish between the RNP, eluting between 7 and 14 minutes, and then the free Cas9, eluting between 15 and 20 minutes. When we complex the lower purity guide RNA with the Cas9 protein and run it on cation exchange, we see that we've lost that ability to separate the two. And so we thought that this would be important information for our clinical researchers when they go about thinking about the reagents that they use to move to the clinic and the quality that these reagents need to be at. Now that we'd established an assay for reliably detecting free Cas9, we then moved to the other side of the coin to look at assaying and quantifying free guide RNA. And so we ran after looking at several methods, we settled on a native page gel as the best way to do this. So when we look at the gel on the left here, this is RNP run on this native page gel at increasing guide RNA to protein ratios. When you look in the first column, this is a 1-to-1 ratio. So the upper bands represent the guide RNA complex with the protein, so the RNP running slower in the gel. And then at the bottom, at the 1-to-1 ratio, we see very little guide RNA, as might be expected. But as we increase the guide to protein ratio up to 1.5, we see more and more free guide RNA becoming prevalent. And then this is, again, to be expected. And then you see that when we do a Western blot using a Cas9 antibody, that it shows that the RNP is migrating at the top of the gel, establishing that everything at the bottom of the gel is indeed free guide RNA. And so we found that this was the most useful and quantitative way to be able to assess free guide RNA. Now that we had established assays for quantifying the RNP and assessing the amounts of free guide RNA in Cas9, we then moved on to what we thought would be a very important study, which was to determine the long-term RNP stability and storage conditions. So as I mentioned, there's little to no data in the literature on this. Most protocols call for the RNP to be complexed immediately before addition to cells. But if our goal is to treat RNP as an off-the-shelf drug, then it's extremely important that we understand how it's to be stored and the effects that storage has on its efficacy. And so we picked 4 storage temperatures, -80, -20, 4 degrees, and 30 degrees Celsius, and looked at them for 6 months at all 4 conditions, and then up to 2 years for -80 alone. So what were our results? Here are the 6-month cutting activity results. And so, again, same gels, same linearized plasmid target. And so let's just look at this first lane of cutting, the 4th lane to the right. At the -80 degree gel, we see robust cutting activity representative and comparable to what we saw at time point 0. We see nearly identical cutting at -20 and at 4 degrees Celsius. And as might be expected, when we move to 30 degrees Celsius, we see a significant decrease in cutting activity of the RNP, likely attributable to degradation at that temperature. When we then go to look at the RNP on analytical cation exchange, we see that at -80, we get a very nice, robust peak corresponding to the elution time point of the RNP, and very similar to what we observed at time point 0. We see a similar peak at -20 degrees and 4 degrees, but then again, as might be expected from the activity data, we see a real degradation and ablation of the RNP complex when stored at 30 degrees Celsius. So no surprise there that, again, 30 degrees is not an ideal storage temperature for a protein RNA complex, but important to establish nonetheless. Additionally, important information to see that little degradation takes place at -20 or 4 degrees as well. We then moved on to our 12-month assay and looked at specifically -80 stability as based on cutting results. And so here on the left gel are the activity assays for our in vitro assay at 6 months, and then the same exact assay at 12 months. And so again, looking at the first lane here at 6 months, you see robust cutting taking place, and you see identical cutting taking place at 12 months, indicating that there is no diminishment in RNP activity. When we then run our analytical cation exchange columns, looking at RNP complex presence at 6 months and 12 months, we see that there is really no degradation at all, and that the results are comparable. So this was great to see. It indicates that RNPs are stable for at least up to 12 months, indicating that they can be stored for this time period and treated in a off-the-shelf drug-like manner. And then we concluded by looking at free guide RNA at 12 months. And so this top gel represents the 3-month time point at all 4 storage conditions. The first 7 lanes are a free guide RNA titration. So this is guide RNA alone, starting at a high concentration and moving to a low concentration. And then the top gel, the 4 right bands correspond to each temperature. So the first lane is -80. Here you see a very clean, tight line of a band for the free guide RNA. You see a comparable band at -20 degrees, but then when we move to 4 degrees, we begin to see some degradation products popping up. And then those degradation products and the guide RNA, the free guide RNA itself, seem to be completely degraded when stored at 30 degrees Celsius for 3 months. We then moved on to look at the same assay for -80 up to a year. And so in the bottom graph, in the bottom gel here, you see the same guide RNA titration. And then in the 3 lanes to the right, you see replicates of -80 degree RNP storage conditions. And the RNP, the guide RNA bands showed no degradation products and look as tight and clean as they did at 3 months, indicating that the free guide RNA is stable and does not degrade when stored at -80 degrees for 12 months. So again, this is extremely encouraging. We intend to conduct these studies for up to 2 years at -80. And so I look forward to getting to present those results in the future, and hopefully they will indicate that RNPs can be stored for longer term with no deleterious effects. So in conclusion, as I mentioned, Aldevron has really made an effort to address the clinical CRISPR space over the last several years, beginning with in the CRISPR space, Cas9 proteins at GMP quality, and now offering this RNP complexing characterization service. So really filling a needed gap for therapeutic providers in this space. So I'm proud to be a part of this. If you have any questions about our protein or RNP offering, please feel free to contact myself or one of my Aldevron colleagues. Until then, I appreciate your time and thank you for listening.